I'm going to record this, but you guys on Zoom, you know what? Let me see. You know what I'm going to do? I'm going to go get my laptop, you guys on Zoom, and I'll put it next to somebody here so you guys can follow along. Let me go do that. They're on your bench there. All those tubes. See, A, B, C, or D? Those are the unknowns. So you just have to take 1 mil out of that and put it into a new test tube. Oh, don't you guys need a Sharpie? Here's one. There's a Sharpie. Feel free to just write on the test tubes. Just write it kind of small because we'll wash it off with ethanol. You guys need something to mark your tubes, no? Right? You want to be able to label these so you know. I mean, unless you're going to keep them in order, but I would suggest labeling them. Let me get you a Sharpie, okay? Okay. Oh, I just went. You know, it's one more time. Right? That's not. All right. We need I need one more Sharpie. Yeah, all you guys are writing, all you're doing is completing the table essentially, right? Yeah, you put ABC on there just so you know what's what, right? Just write it on it. Yeah, you can write right. Just write right on. What you guys mix it with?
What do you mean? Oh, I just pick it up and slosh it around, I'm assuming?
Yeah, just give it a little swirl to mix. You know what? I'm gonna leave this here. Just update. Give an update of each test that you do on the camera for me, somebody. Brad, you guys, when you get more time.
Sure.
There you go. This is ginormous. This might be good enough. I just didn't know. I'm not gonna get my laptop. Yeah. Just open that up. Yeah. So you have you gotta do each channel. 5 drops, swirl to mix, and then look for the color change. Okay? How you guys doing? What's up? So you guys want to put for each unknown, right? A, B, C, D. But the easiest way to do it is have A, B, C, D in here, right? Your 1 mil of each one, and then take all 4 of these tubes and do the Benedict test, right? And then you're gonna write down for A, B, C, D what happens with the Benedict test, right? What color it is, and then if that's positive or negative. So remember, it's positive. So, or you can do iodine first, whatever one you want to do first. So if you're gonna do Benedict, it's in the middle.
Yeah, that's what I was saying. Which one? Where's
that's Benedict, right here, and you have to boil it, okay. Right? So here. Okay, so you're gonna do Benedict first. But I would set up all your unknowns and do them all at once. There's no sense doing them one at a time, right? So you can bring your whole rack of tubes over there if you want. Just make sure you have all your unknowns in there.
I'm afraid.
You good? You don't think you good? Okay, it's all right. They should say. Usually use a smaller beaker, but I think they did it. Oh, see, like you can already see that one's already starting to change, right? Well, be careful with the bottom. Yep, 5 drops into each one. So you should have all of your unknowns here, right? B, C, and D. Put them all in there. Okay. Do them all at once, right? Then you put them all in there like at once, like project. Yeah. So just update the camera there. I'm recording it when you're.
Do you want to bring it over?
Yeah. How?
Oh.
Here, let's slide this over.
Yeah.
Let's not light anything on fire. Right?
Pull that over here.
Yeah. You guys got it now?
Yeah, that's max.
Yeah. Okay. Oh, that's probably good enough.
Do you want me to angle it down?
Yeah, probably. Can you see what's going on there? Here, let's unshare the screen and put the camera big. That camera's actually halfway decent, so let's see. Let's pin this. Yeah, there you go.
Look at us.
Okay. Sorry, the dry doors. I don't have no more Sharpies. Sharpies are like the hardest thing to come by. They always have. Here. Oh.
They're chilling.
Here.
Do I just pluck them out?
You all right with that one? You want? You sure? It's fine. You only have to do a little. Yeah, like 1 to 2 minutes. Honestly, you can pull them out. Just be careful. Here, you can use these things, but fingers are better, to be honest. It won't gonna burn yourself. Probably not really good lab safety technique, but
Okay, it burns not.
this thing is. These are worse because they like clip it and then they break the tube, so it's actually more dangerous. All right, so there you go. Super easy. So you got one of them.
Let's see.
Right? So just kind of, yeah, you got it. Just look at the color for each one.
This is A. This is B.
Yep, good.
We got C.
Thank you.
No problem. Good old fashioned D.
And D, good. So that's for the Benedict's test. That's all you need to tell them. They figure out the rest themselves. That's interesting that two of them changed. I think I know what the unknowns are.
I'm assuming.
Well, you know what they should be, so.
Just the color, right?
The color, and then if that indicates positive or negative.
Okay.
Actually, yep. Let me just do something real quick. It's actually a good one. Nope. Yeah, so like if you guys look here, right, there's your Benedict's test.
Okay, I got purple for A.
Yeah, so the purple's a little iffy, just so you know. Right? It'll be kind of green, yellow, orangey, or red. I don't know why this one's kind of turned that way, but whatever. Just note that as your color change, and then you can make a deduction from there. Okay. Yeah, so I would that's definitely just a negative test, right? Like yellow or whatever. Yeah.
Okay. Which is negative as well?
Yeah, so are you guys calling the purple one? Oh, you did yours for protein. So perfect, right? You got, now be careful because you only want to do for, is that for your Benedict?
Yeah.
You didn't do that. Okay. So that purple, I'm not so sure I would call that positive. To be honest, usually it doesn't turn, I mean, if you look up there, really your positives are here, green, yellow, or red.
Oh, okay.
So I would call that not a positive test. It usually doesn't change color at all, but if you notice, do you like see how clear it is compared to how thick? Yeah. So that's negative, that light purple, okay?
Yeah,
that light purple for Benedict's doesn't indicate a positive test. Usually, like I said, it doesn't change at all. But do you notice like that orange one kind of looks like thick, and then the purple one's like clear. So so which one's your positive there? D, okay? Yeah. Call all the other ones negative, okay? Yeah. So the light purple, for those of you guys on Zoom, for the Benedict's test, a light purple, I would call negative, okay? Right, I'm going to share your.
Want to do 2 and 2?
Oh, yeah. Let me just share the screen real quick, just so that I have that on the video as well.
we have to use a different one of these each time?
Yeah, I mean, I wouldn't mix stuff with it, yes. So I guess the answer is yes. What happened to my, what did I do? Down here somewhere.
I am. The last one. Go ahead, you start.
All right, anyway, let's do that again. Okay, so for the starch test or the iodine test for starch, this is what A looks like, light yellow. B is an amber color. C is dark purple, dark blue. And then D, same thing, is amber. So you should be recording these. You're trying to determine what the unknowns are.
Professor, can you put it again, the colors, please?
All right, I'm gonna turn off one of these. You guys are done with that water bath, right? Yes. Yeah, you don't only need to boil for Benedict's.
Yeah.
Right? Yep. And then when you, for beer, did you guys do beer yet? No. Okay, so just remember, just add it under the hood. Bring all your tubes under there, just add it, and then it's instantaneous. That's it. You add it, swirl, and then you're done. It's super quick. And then we'll just confirm what you think what is. I'll write it on the board. All right. Yeah. Oh, you guys are sharing. Yeah, that's definitely a negative.
So how do you say that one?
Yeah, so this one, because of the what's in there,
Oh,
the
what's
solution is
inside.
like a little, yeah, it's just a little more cloudy too, so I think that's why I just.
Okay. So the one you say, black.
Yeah, black. What you use black?
Okay. A is negative, B is negative, C is positive, D is negative. Yellow, this is going down, it's yellow, and it's black.
You added this already, right? Guys, what did you get so far?
So far we.
Well, yeah, so you, right, so
Oh,
your
they're up
possible
here,
choice,
okay.
choices are there, there's possible choices at the,
Okay.
yeah, although you probably could make something up, probably came up with it. You guys are on Zoom, you guys are gonna need, you need to know the colors for each individual tube or each individual test for each unknown. You know what, I'll grab them out of there. I don't want anyone to get burned. I should be, there's clamp, there's like let me grab them. If I get burned, it's okay. If you guys get burned, it's not cool. You're good, you don't need to leave them any longer. B. So this is, I'll just do it again. Unknown A, this is for Benedict's.
I have to do it again.
No, you're fine. Oh, I'm just showing them. This is D for Benedict's. And then C. Right? And then what's this guy? This is B. So B, we're going totally out of order, but whatever. C, this is D, and this is tube A. Okay? So
Professor,
this
can you
one,
move them closer
the
to
Benedict's,
the,
that purple
the
color,
camera?
it's not a positive test for Benedict's, right? So the Benedict, I had the picture up there before, but Benedict's would be either orangey red or green. So you can you can figure that out, which one's negative and which one's positive. Let's shut this off so that we don't, that's like my worst fear, that I'll leave the hot plate on and we'll burn down the building.
So the iodine test is blue red, right?
Iodine, correct. If the iodine turns blue-black, that indicates that starch is present, right? So that's your positive, right? And then you just need to determine in that conclusion column what's what. In the top of your form, it tells you what are the possibilities. Water, I think it's water, albumin, starch solution, and glucose solution, right? What do you guys figure out?
It's the albumin solution, the water, starch.
They're pretty easy. So you know what? Write it down on the board.
You want to write all of them or just one?
You can write all of them. We'll just see if we all agree.
Yeah, I hope I can spell.
Yeah, I can't spell those. So for starch, this is tube A for the iodine test for starch. This is tube B. This is tube C. Hey, what's up? This is tube D. Are you done?
Yeah.
All right, so just wash it there. See this go here?
Yes.
Yeah. This or this part and this goes. Okay. You know what? Don't wash it there. I lied. I probably wash it. Over here. That thing.
Professor,
Yeah.
what color is it?
I mean, I would call that, yeah, that's for starch. That's like bluish, dark blue. Yeah. You could, look, I mean, if it doesn't get dark, it's negative, right? But if you see it get darker, then it's a positive test. All right, so this is the burette test. Again, we should know what that's a test for, right? Tube A, tube B, tube C, and tube D. So in your table, you're noting the color and whether that's a positive or negative test, and then determining which solution is which. Unknown A, unknown B, unknown C, and unknown D.
Professor, can you show them again, please?
Well, once you're done and see if you came up with the same conclusion. I shouldn't have told you that, Figure it out first. Where you guys at? Did you do all your tests?
Yeah, we won this conclusion.
Yeah, right. So now based on D, these are your four possibilities here. Right? So we need to determine So no, you're writing positive or negative here in the same column, right? So like if it turns orange, it's positive. So instead of positive, that's fine. Just come up with so your conclusion is what it is. Okay? Yeah. So just add that in. Yeah, that's fine. All right, then those of you guys on Zoom, in the conclusion, again, this is all in the document, but in the conclusion, you're writing what it is. Is unknown A water, starch, glucose, or albumin? Is unknown B one of those? So it could be one of the four. And there's each of one, right? One is water, one is albumin, one is starch, and one is glucose. Again, that's clearly outlined or listed in the intro to this, okay? Once you guys here are done, you're free to go, right? So do we all remember? So we're going to need to know these
Okay.
for the lab practical, right? So even though it's electronic, like I'll put a picture and be like, is this a positive or a negative test result, right? So you gotta know what the tests are for each type of biomolecule, and then what indicates a positive or a negative, right? Okay, I mean, that's essentially what you guys did, what we did in the other lab too. Right, so if you're finished, just rinse all those out, and I'm gonna put a, did I already do that? Yeah. So, and then just put your rinse tubes upside down on this rack, okay? Thank you. I wonder what happened to the phone. Just so that we have you here, remember for Benedict, for you guys that are on Zoom, Benedict's test possibilities are there. Again, it's in your, right? So those are the possibilities on the screen that I'm sharing in terms of it being positive versus negative. Yeah, you can throw those out. All those little plastic pipettes you can throw out. Right, so for tests for starch, here's your negative test, here's your positive test. And then we did one other test, and that was the Biuret test for proteins. There you go, negative test and positive test. All right, what'd you guys get for your for the unknowns? Yeah, I need those back. Thank you. Make sure your name's on it. That's my record of who was here. Thank you. Thank you. See, quick and easy. What did you guys get? So how come you chose this guy as being, all right, well, we already have some, so for Benedict, the purple isn't positive, right? Remember this, that was, like color, okay. So for, right guys, so for unknown A, it was negative for Benedict, right? Positive for purple, I mean positive. Positive for Biuret, but negative for starch, right? So that indicated what for Biuret? That it had what in it? What's your Biuret attached for? Yeah, protein, right? Yeah. So albumin, a protein. So there you go. For unknown B, right? So now for unknown B, the only way you would determine it's glucose is if for the Benedict test, it was positive, right? So look, for unknown B, blue, that's negative, and iodine, if it's yellowish, that's also negative. So it's negative for everything. So which one up here would be would test negative for everything? Water. Right, so what's your unknown B? Water. So now for unknown C, right? No, what did you get as a positive test? So this is negative for sugar, right? So this is sugar. Negative for protein, but positive for what? Iodine only tests for starch, right? So it's starch, right? Unknown C is starch. For unknown D, right, you had a positive Benedict test, which is your test for sugars, simple sugars, right? These are all negative, right? So what's unknown D then? Right, unknown D is glucose, unknown B is starch because, unknown C is starch, right? You gotta do that. You want another one? Yeah.
I'm like,
Right, do you guys understand? That's why I'm going through it. I want you to understand why you're choosing that is based on those tests. So here, you want to do another one? Yeah. Oh, sorry, I'm giving you Brad and Jason. Okay. Yeah, just redo it and let me know. So again, those of you guys that are on Zoom, you're determining what the solution is for each one, unknown A, B, and C, based on the tests that we did. Okay, so I showed you guys the colors for each of those 3 tests. You have to determine what it is. Is it water? Is it starch solution? Is it glucose solution? Or is it albumin solution? All right, you have yours. You finish yours? Yep. Right, so you understand why, right? Yeah. You're just looking across here. So because this guy, the only positive test you had was for protein, right? Yeah. It's albumin. Here you have all negatives. It's water. For C, right? The only positive test you had, starch. And then here, this was for Benedict's. You had a positive, right? So that's why it's blue.
yeah.
Okay.
Thank you.
Yep. All right, so I'm logging off those you guys on Zoom. So you have all the info that you need to determine what's what, right? So you need to determine what each unknown is based on those tests that we did. Okay? Bye.
Was this, Professor, was this being recorded so I could go back? Because I missed a couple of answers. Thank you.
Thank you. Thank you. Does anyone have a question on Zoom?
I do, Professor. I do. I have a question.
You're muted. What's up?
I have a question.
I don't know why I'm not hearing you. Wait a second. I know why. All right, go ahead.
What are you gonna post?
I mean,
this
you
so I
can.
go back and see because I'm missing a couple.
You can throw them away. Yeah. Katya,
I'm missing a couple of solutions. Were you recording the lecture or the lab?
Go ahead. Sorry. Go ahead again.
Were you recording the lab? Because I'm missing a couple of solutions.
Yeah.
So you're gonna post it right after so I can submit it?
I'll post it when I get a chance later tonight. Okay.
All right, no problem. So I can submit it by the end of tonight.
Just check. Yeah, just check Moodle. It should be up. If I have time, I just have to get out of here, but I may be able to post it right now. But I gotta wait for the video to be ready. It has to process first, so it might just be a little later. Okay.
All right. Okay. No problem.
Okay. No, but I should have your test